Welcome to the Explainer. So, have you ever wondered what it actually takes to turn raw crystallographic data, or just endless strings of PDB coordinates, into a spatial reality you can actually see? Well, today we're looking at a tool that is essentially the ultimate magic lens for scientists. It's called PyMOL, and let me tell you, it is absolutely revolutionizing how we interact with the molecular reality of biology.
Because think about it, the true challenge in modern drug discovery isn't just knowing that a molecule is out there. The ultimate barrier to curing disease is that these microscopic molecules are completely invisible to us. So how do we bridge that gap? How do we take angstrom-level coordinate data and turn it into a visual model that our human intuition can actually grasp? To spot those hidden, cryptic binding pockets and understand structural vulnerabilities, we absolutely have to bridge the massive gap between chemical reality and what we can actively analyze on a screen.
And that is exactly where PyMOL comes in. Created back in the early 2000s by Warren Delano, this is an open-source visualization tool built specifically to give us high-quality 3D images of biological molecules. You see, Warren was a huge champion of open-source science. He built this tool to democratize structural biology before his tragic passing in 2009. And his vision? It was a real game-changer. He basically took those endless strings of abstract, confusing data and transformed them into a vibrant, interactive 3D map of life's fundamental building blocks.
Okay, let's dive right into this roadmap. Our journey today goes from single molecules all the way out to global drug discovery. First up, visualizing proteins and DNA. Then, exploring complex molecular machines. Third, accelerating drug discovery. And finally, education and accessibility.
So, starting with part 1. Visualizing proteins and DNA, the fundamental building blocks.
Now, focusing in on the microscale, the real workhorses of cellular function are proteins, and they have incredibly complex three-dimensional structures that dictate exactly what they do in our bodies. PyMOL lets scientists actually visualize these proteins using ribbons, sticks, spheres, and surface models. But here's the cool part. Scientists don't just sit back and look at these shapes like they're some static painting in a museum. No, they actively manipulate them. By just toggling from a ribbon view right over to a surface model, a researcher can instantly explore active sites, protein folding, binding interactions, or even uncover hidden hydrophobic pockets that are completely invisible in a standard textbook diagram.
And it's not just proteins. Visualizing DNA in PyMOL reveals its helical nature, the base pairing, and the atomic stacking. And this brilliantly illustrates why this tool is so vital. It gives us the precise major and minor groove widths. Mapping out these basic genetic structures in 3D is absolutely the crucial first step towards studying genetic mutations and ultimately repairing mutated DNA with custom gene therapies or CRISPR targeting.
Which leads us nicely into Section 2, exploring complex molecular machines, zooming out to biological cities. Because what happens when thousands of these fundamental proteins and nucleic acids start working together?
Now, what's really interesting about this slide is how we categorize scaling up. We are transitioning from a single protein or RNA right up to massive biological assemblies. For instance, the ribosome is basically the factory floor for protein synthesis. Then you scale up to genetic material like a virus, which infects host cells and replicates. And zooming out even further, you get complex organelles like mitochondria and chloroplasts handling cellular metabolism. PyMOL lets researchers dissect these massive, complex biological interactions right at the molecular level.
By dissecting the microscopic machinery of things like ribosomes and the infectious structures of viruses in a 3D space, researchers can actually pinpoint the exact mechanistic weak points. And again, we aren't just looking at static pictures here. Researchers are using this interactive map to actively understand how a virus infects host cells. They're watching how a virus undergoes conformational changes, which is exactly what lets them build targeted, highly specific antiviral drugs that literally jam up that machinery. Now, let's transition into the highest stakes application of this software in Section 3, accelerating drug discovery, designing the cure.
At the absolute heart of designing a cure is a process called molecular docking. Basically, this is predicting the preferred orientation of a small molecule when it's bound to a protein target. You can think of it like finding the perfect key for a lock. But it goes way beyond just a static fit. It's about visually understanding specific binding affinities and evaluating energetic favorability. PyMOL lets structural biologists really dig in and analyze things like steric clashes and hydrogen bonding networks, ensuring that the key actually works and the fit is energetically sound.
Let's move to the next concept and see how this all builds together into iterative drug design. It's a rapid, three-step loop. Step 1. Visualize the interaction between the molecule and the target. Step 2. Modify the chemical structure, visually right there on the screen. And Step 3. Predict the new binding affinities and pharmacological activity.
If a drug candidate doesn't quite fit, you just tweak it in silico. Literally adding a single methyl group visually can mean the difference between a totally failed compound and a life-saving blockbuster drug. It's an incredibly dynamic process that shaves years off development.
Finally, we arrive at Section 4, Education and Accessibility, opening the lens to everyone.
Shifting gears from high-level lab research, let's look at how this tool democratizes science. We have open-source PyMOL, which is completely free to use and highly accessible for students and global researchers. On the flip side, you have PyMOL by Schrödinger, a proprietary version offering additional features and paid support. So the crucial point here is that PyMOL scales perfectly to your exact needs and budget. It bridges the gap from a free, vital educational resource all the way up to a high-end enterprise tool driving multi-billion-dollar R&D pipelines.
And it sits inside this massive visualization ecosystem. PyMOL is truly cross-platform, Windows, macOS, Linux, you name it. Plus, it has this thriving support network with active online community forums sharing Python scripts to automate rendering. Now, we should definitely acknowledge that there are some great alternatives out there, like Chimera, VMD, or JMOL, which are fantastic depending on your specific niche. But for education, PyMOL is just incredible at teaching those super complex 3D spatial relationships that are so foundational to modern biochemistry.
As we bring this explainer to a close, I want to leave you with this quote. PyMOL continues to play a critical role in advancing our understanding of the molecular underpinnings of life. I mean, just think about how far we've come. A couple of decades ago, atomic coordinates were literally just numbers printed out on a sheet of paper. Today, we are holding the 3D architecture of life right on our screens. We're spinning it, analyzing it, and interacting directly with the very fabric of biology.
Which brings us to one final, huge question. With the power to visually map the machinery of life, what invisible cure will we design next? The tools are literally right at our fingertips, and the future of drug discovery and molecular biology is just wide open. Thank you so much for joining me for this explainer, and keep exploring.